Hong Liu, and I'm a professor in chemical and biomolecular engineering at Georgia Tech, Georgia Institute of Technology. We are in the city of Atlanta in the US. As you can see my background, this is actually our really beautiful campus. I direct a lab that uses microfluidics and automation and quantitative biology, quantitative microscopy, and data science to try to understand biological systems. So we typically do our work with a genetic model system, C. elegans, which is a nematode. We also have other projects that are also working with cultured cells, organoids, and other developmental systems. We're very interested in the nervous system in general, but also other problems in biotechnology and cancer and cell biology. So my lab actually does a lot of work in a clean room, as well as a wet lab, which my lab is actually going to show in the rest of the video.
Here we are in the Marcus building, which is where we do our microfabrication in the clean room. This is one of the largest clean rooms in the southeast. You can see here a map of the different equipment where we only use the red section there for soft lithography. And you can have a view here of the equipment in that A, here where we have the optical lithography tools, and then a little bit further down we have the spin coating, and the hood. After we fabricated the master in the clean room, we are now here in our lab where we make the device using our PDMS station. One application in our lab is for cell biology, and we have dedicated a whole room separated from the rest of the activities to maintain a sterile environment.
So this is our general lab space where we do all of our worm culture and basic biology. One of the questions we had as a lab was asking how do C. elegans age differently based on their environments. So we've created microfluidic platforms like this one specifically designed to image aging populations and study those types of questions as well.
So here is our microscope view. So here we have 6 distinct microscopic setups, including bisectoscope, inverted fluorescence microscope, projectorscope, and light sheet microscopy as well. So here is one of our most specialized equipment. This is the projectorscope. So we use the projector as a light source, and we mount the microfluidic device on the stage and image the behavior tracking, and also do the fluorescence functional imaging as well. And also we use this pressure box to control the microfluidic device as well.
One of the facilities that we have at Georgia Tech is state-of-the-art confocal system. Combining these confocal systems with microfluidic techniques, in our lab we are trying to answer some interesting neurobiological questions in C. elegans. And one of the other research that we do in our lab is to develop some machine learning algorithms that will enable biologists to extract meaningful information from these microscopic data so that they can focus on their biological question of interest. µTAS is a huge conference that always supports and encourages emerging fields like machine learning, and we are happy to share our machine learning research at µTAS.
I have been a PI for 16 years, and I have always grew up with the microfluidics community. When I was a student, I attended µTAS several times. And so it's very exciting to see that my students now are interested in attending the meeting. The subject area for µTAS has been ever expanding, and we're very excited to be able to share our work in using microfluidics for biological, medical, and data science applications. So hope to see you at the meeting.
Hello from South Africa. My name is Paul Watts and I'm based at Nelson Mandela University in the city of Port Elizabeth. The overall vision of the research that we are conducting is to be able to synthesize APIs and drugs for the continent of Africa with the vision of producing them at lower cost in order to enable the healthcare provision of the continent to be improved. The principal way that we are trying to achieve this is utilizing flow chemistry where we start the research at a pure microfluidic level and then move up to larger scale flow reactors in order to facilitate industrial drug manufacturing. What I'm going to do now is to take you on a walk around the laboratory to show you the facilities that are available. The laboratory, let's show you some of the research equipment. So this is a traditional organic chemistry laboratory fundamentally, and where we always start is using these very small pure microfluidic reactors where the channels are a couple of hundred microns in diameter, and these have volumes of about 10 or 20 microliters. So the way that we actually operate them is they fit into these standard holders that you see here. So we just pump the reagents in using a couple of simple syringe pumps. Here you see two of them connected in series for multi-step chemistry. Using this type of research, we develop all the fundamental technology and processes in order to prepare various AIDS, TB, and malaria drugs and diabetes drugs. And then I'll show you in next step how we actually scale the research up to a larger scale. So I just told you about the pure microfluidics. Now what we do when we want to scale reactions up is to move what I call mesoreactors or something like that, which is probably not something that my µTAS colleagues would use that often. But what you see here is a reactor from the Little Things factory in Germany, which is the sort of size of a cell phone, has sort of internal volumes of 1 or 2 milliliters, and it enables students to sort of scale up their work to maybe gram scale synthesis. The way that you would actually use this is just to immerse these reactors in an oil bath or a coolant bath and pump the reagent through using solvents in exactly the same manner. If you wanted to prepare a slightly larger scale, you've got here sort of a reactor which is about 15 centimeters square, which enables you to go to a slightly larger scale. But again, the key is that you've got sort of intense mixing, so you've got very, very efficient ways of actually doing synthetic organic synthesis. And once you've done all of your proof of study work, what we've got over here is the Kiloflow system from Chemtrix. Now, rather than using a syringe pump to pump your reagents into the system, you use small HPLC pumps. And this system here has flexible volumes ranging from sort of 10 mil to 100 mil, something like that. And we just finished a case study for a partner where we sort of prepared 100 kilograms of a diabetes drug in a week. And 100 kilograms is a huge amount for a typical university laboratory. So what I've tried to show you today is the concept that we are working on at Nelson Mandela University to prepare drugs which will hopefully enable people in the region, in the country, to have a better quality of life going forward. Thanks very much.
Hello, welcome to the Micro and Nanobioengineering Lab, part of the Biomedical Engineering Department at McGill University here in Montreal, Canada.
Hi, I'm David Juncker. I'm the PI of the Micro and Nanobioengineering Lab, and so our lab develops new micro and nanotechnologies for the advancement of human health and to advance our understanding of biological science. So in our lab, we are a diverse group of scientists and engineers from biology, physics, and all different disciplines of engineering, working on maybe 3 main aspects. And they are lab on a chip with our capillary or capillary microfluidics. And then we have bioanalytics side with liquid biopsies as our focus, analyzing multiple biomolecules. and finally driven by our 3D printing efforts, tissue engineering, and organ on a chip. So I wish you a lot of fun in this virtual visit and hope to see you at µTAS. Thank you.
In this video, we're going to show you around the lab facilities and highlight some of the work that we do. This is our mechanical tools room where we keep all the screws, drills, woodcutters, and other tools that we use to build some of our experimental setups. Next is our cell room. It's biosafety level 2, and this is where we do routine maintenance for our cell cultures, engineer biomimetic 3D tissue models, organ-on-a-chip devices, and cancer co-culture arrays. These are the Microfluidics benches. We have a paper cutter, 2 commercial 3D printers, and an inspection microscope. The printers are heavily used to fabricate chips and tools for so many applications, including diagnostics for COVID-19, organoid vascularization, cell aggregation, EV detection, and biofluid sampling. Moving to the next section of the lab, here we have a laser cutter, plasma chamber, and multiple chemical fume hoods. Next, let's go to the proteomics benches. Here, we have a multi-mode fluid dispenser and all of the freezers for the lab. Here we have our precision spotter used to detect and characterize extracellular vesicles from various biosamples. The spotter is capable of dispensing droplets in the picoliter range, and we use it to create antibody microarrays to capture EVs. Next is everyone's favorite place during a hot summer day. It's the cold room where we store samples and stock some reagents. Finally, let's go to the dark microscopy room. We have 1 fluorescence inverted microscope and 2 confocals that we use for imaging and analyzing our results. We are keen to join µTAS every year to connect with the microfluidics experts from around the world and present our work in progress to the scientific community.